Okay, so now for a few recommendations, and I think you'll find that most of these are just common sense, and you're probably doing these things now. But here's some considerations to think about before the crop cycle. All the contact surfaces must be cleaned and disinfested. And cleaned means getting rid of all the organic material, because disinfectants, no matter what they are, will not penetrate into this infected plant material and clean it up properly. We're going to assume that the initial water coming into the system is clean, unless, of course, it's drawn from surface water. Then all bets are off. Pythium is common in surface water, and Phytophthora occasionally occurs. We certainly can't assume that the incoming plant material is clean. If you're propagating this yourself, you might be doing a good job, and you may have a high level of confidence that it's clean, but many of you are bringing plant materials in from another greenhouse, and it's really hard to tell if you're bringing in any pathogens or not. So a survey or some scouting should be done, and discard trays with stunted, wilted, or yellow plants. Discard the whole tray, because if you have root rot in some flats, then other the plants that there's plants there that are infected but not showing symptoms yet, and we simply do not want to bring these things into our production facility. We need to restrict access to the flood floors. Only designated people are allowed to go out on flood floors, and with proper footwear, clean, new footwear, booties or some other footwear. Foot baths may or may not work for you. Foot baths can become contaminated and then become a source of the problem, especially if they're not changed recently. And it's not clear that even fresh foot baths work well. There could be other ways to get around this, and that is using fresh booties before you go in. One of the problems with something like foot baths is that they can give you a false sense of security, and perhaps your shoes are particularly dirty and don't become sanitized properly in the foot bath. If you've had a history of a problem, you should plan on using preventive fungicides for root rot. Okay, cleanup after each crop is finished. Vacuum or sweep all plant debris from floor. Disinfect all surfaces in contact with flood, water, or plant material, just as we did before. Now, we have to clean all the corners and other places that are kind of inconvenient to reach with power cleaners, and that means cracks and crevices as well. If you have filtration systems, they must be cleaned. Don't forget that filtration systems may capture a lot of those pathogens, and then when a lot of them gets in there, they'll start bleeding through into the system. Change your foot bath disinfectant if you're using one. Okay, so here we see a power cleaner, and what I want you to see in this picture are a couple problem areas outlined in yellow here. The one in the lower left-hand corner is not cleaned by the circular brushes, so we need to go in there with a hand brush and clean that area. And the cracks, those cracks actually might receive a lot of the organic material that we want to get rid of, and they could even get packed down in there. We need to get a power hose and blow the stuff out. Clean up following disease. Empty water tanks and remove all organic material. That means you can't have mud and junk at the bottom of the tank, because that mud and root pieces and so on are simply smoldering embers that will allow the fire to come back again after everything's put back into place. Once it's clean, it can be disinfested. Clean all floor, bench surfaces, and disinfest. So we need to use pressurized water to force plant material out of those cracks that we looked at and the nooks and crannies. We need to flush plant debris from the distribution system. And some of the older systems, this could be very difficult to do because they may dead end under the floors. Remember the rule that all surfaces must be cleaned before disinfestation. Starting up after cleaning, fill water tanks and add an appropriate oxidizing agent. Turn the system on to distribute and allow to drain. Let's not recontaminate the system. We have to make sure that incoming people, equipment, and plants are not carrying contaminated soil or plant debris from the previous crop. If you're under new construction, I have a few suggestions for you. The water distribution pipes that are under the floor should have round corners and no dead ends. We want to be able to clean these things out easily. They should have cleanouts to facilitate flushing. You should compartmentalize a range so that regions are serviced by separate tanks with closed distribution systems. This way, if you run into a problem in part of your range, it'll be compartmentalized to the distribution system, to the plants that are receiving water from that distribution system. And so it's sort of like a firewall. Consider installing ultraviolet water cleaners and water filters. And be advised that ultraviolet water cleaners will not work in dirty water. And also the flow rate has to be at a specific rate in order to properly clean the water. There should be a dedicated space for all incoming workers to change into clean footwear. And I think this is a better idea than the foot baths. I'll share a couple of experiments that we did. We're concerned that peat moss or just growing medium would shorten the length of time that these oxidizing agents are active, and it wasn't too bad actually. But we used Fusarium Chlamydospores as our test subject, and we had started out with 70,000 of them. And then we took our oxidizing agents and we exposed them to organic material for 5, 10, 20, and 30 minutes to see how effective they were after that pre-treatment with organic material, to see if they lost much of their power. And we had our control had no exposure, and 0 plus 0M was just a brief rinse through of the peat moss. So at the top is a control, and that should be a straight line, and it is pretty much. The second one down is 500 parts per million of hydrogen peroxide, and that held up pretty well. The 50 parts per million of chlorine dioxide did not hold up that well. It actually lost a fair amount of activity in 30 minutes. And the 500 parts per million of xeritol held up quite well, and also killed a lot of the Chlamydospores of Fusarium. So you can also see the effectiveness of these materials. Now 50 parts per million was used for the chlorine dioxide because that material can be phytotoxic at higher levels. So we did 2 trials actually, and they came out pretty similar. There were significant differences between the Chlamydospore viability and the treatments. And trial 2 is similar to 1, but the hydrogen peroxide was not effective in the other trial. And the sphagnum moss only affected the chlorine dioxide. The other materials held up pretty well. Doing some work with the effectiveness of chlorine dioxide and against various propagules of Pythium fusarium and then Erwinia bacterial cells, it only takes 1 to 5 minutes to kill Pythium zoospores at a rate of 1 part per million. Now, in that experiment we just looked at, we were using 50 parts per million. But we only need about 1 part per million to kill zoospores. The zoospores are pretty sensitive. Fusarium spores were eliminated after 20 minutes with only 5 parts per million. Now they're a little tough. They're not Chlamydospores here. They're simply vegetative spores that Fusarium produces that would likely go through the water system. Erwinia bacterial cells, surprisingly, were more resistant to chlorine dioxide, and it took 20 to 40 minutes and 20 parts per million to kill them. Now, we didn't test Chlamydospores and oospores because experimentally they're much more difficult to work with. But they're going to be much more resistant to these chemical treatments. And at some point we need to look at how resistant they'll be. And don't forget what I told you earlier, that if they're inside plant material, they're going to be completely protected from these disinfectants. And that's all, folks. Thank you.